Hi everyone. Hello. So before let me just say about this, okay. So is your term 2 exams with Unacademy Plus or Unacademy Plus subscription at flat 10% off. So that is with the code Vani YT. Okay. Yeah, I am audible. Alright, I'll just reduce this. Is my code. Okay. So with this you will be getting 10% off on your subscription, that is on plus, so that is on Unacademy Plus. Okay, so before I start, let me introduce myself. I am Vani Kari, biology educator at Unacademy. I am an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree, also a founder of a startup where I grow microgreens by vertical farming technology. Just a second, I'll just change this one, and also your chat is anyway not visible again, so maybe I need to solve this problem once for all. Let's see. Okay, so this one, yeah. So before I continue, so let's see what we are going to study today. So last class the function was I was supposed to mention, so I did only about the ultrafiltration. So let's see all these functions and then I'll move to this constituents of urine and other things. Okay, and this is my code again, Vani YT, that is for 10% discount. Okay, so you can also follow Unacademy Champions on Telegram, so link you will find in the description box. And YouTube now, so let's target about 1000 subscribers. Okay, so you can press on bell icon, and so with this you will be getting the notifications. Just a second. All right. Yeah, so last class we were discussing filtration, so I'll just quick draw this particular structure, that is ultrafiltration now. Okay, then I'll just explain about the functioning with respect to this ultrafiltration, the reabsorption process and everything we will see. Okay, so for now, just a second, I'll change the color also. Okay, so you can see this one. Okay, so there is nephron. Okay, so this one is now a nephron which is having a structure as such. Okay, so this is now a nephron and this nephron is continued as such. Okay, so this will be forming this PCT. So now this one is actually the PCT. Okay, then we have this particular structure. Okay, so this is actually the Henle's loop now. Okay, so please excuse my drawing skills with pen tab. Okay, so this one is actually the DCT. All right, so this one is DCT which will be joining the duct, that is collecting duct. So here we have this collecting duct now. Okay, so this collecting duct at junction, so it is going to collect the urine from different parts also, I mean different nephrons also. Okay, so this is now a collecting duct, so we will just label them, quick label them. Okay, so now this one is collecting duct. All right, so this is collecting duct, then this one is distal convoluted tubule, and this one is proximal convoluted tubule because it is near. Proximal is nearer, so nearer we find it to the Bowman's capsule. Okay, and this particular is Henle's loop. All right, and then what else we have? We have this Bowman's capsule. I'll just label it over here. So there is a Bowman's capsule, and with Bowman's capsule what you will find, so there will be a glomerulus. Okay, plus glomerulus, so together what do we call it? We call it as a Malpighian capsule. Okay, so this one is now a Malpighian capsule. All right, so these are the parts. So I'll just draw this afferent and efferent arteriole, so the structure of this nephron will be complete. Okay, just take the different colors so that I can draw the arteriole. So you know that the renal artery which is coming, so it has to divide many times. Okay, so branching and rebranching has to occur then. So now this one is actually the afferent arteriole. Okay, so this afferent arteriole, so this is going to divide many times, so there will be formation of many of these capillary formation will occur. Okay, so this is completely forming the capillary. Then what is happening? So it will be leaving. Okay, so this is leaving. All right, and you need to make sure that the diameter over here is more and the diameter over here is very less. Okay, so that is why there is a hydrostatic pressure that is built up. Is there a disturbance with my video? Yeah, I hope it is fine now. Let me just okay, anyway, so this is actually the afferent. So I'll just label them. So this is the glomerulus what we have, and this one is afferent arteriole. Okay, so this is afferent arteriole, and this one over here, so this is actually the efferent arteriole. Okay, so this is efferent, it is E. Okay, so efferent arteriole as it is living. Okay, so you know the renal artery will divide many times, it will form the arteriole, then arteriole will divide many times, it will form the capillaries, and here you know this one is the efferent arteriole. So now in this case we need to draw the capillaries again. So let's draw this one. So it is gonna form structure as such here, so this will further divide. So it is gonna form this kind of structure which we call as the capillary. The secondary set of capillaries are being formed here. Okay, so this will further divide again. So then what is gonna happen? So let's see this structure now. Okay, so this one over here, so again it will be forming the capillary set. Okay, so this is gonna surrounding this Henle's loop, so this will be forming the secondary set of capillaries. Okay, so there is secondary set of capillary which is formed. Okay, so anyway this one is too much of gap I have given, so let me just erase certain parts here. All right, so this part I don't want, this part I don't want, this part I need to make some changes. Okay, so we will see the structure now. So these are the secondary set of capillaries that are formed. All right, okay, so now you know, so these are the capillaries that are formed, so we need to join them all. All right, so we are joining them, and here the capillary formation, and here also you'll find this capillary which is surrounding this. You know, this one is actually the descending limb. Okay, so this is descending limb of Henle's loop. Okay, and this one is ascending limb. Okay, so this is actually the ascending limb of Henle's loop. So now what will happen? So all of these are joining together. So this is a blood vessel, so we need to draw two lines over here. Okay, so then all of these are again forming a capillary set. So what will happen? So again they are forming this capillary over here also, because some amount of reabsorption will occur here as well. Okay, so here also some reabsorption. So we will see what is reabsorption and other things. Okay, so now this is the structure. Okay, then these are all joining, and this will be joining where? It will be joining the renal vein. Okay, so it will be joining the renal vein. So here it was coming from where? It was coming from the renal artery. Is this clear now? So this is the structure we have. Okay, so now what we need to check in here is, so we need to understand the ultrafiltration process. So what is gonna happen? So let's see here, the blood is gonna flow. Okay, so you know there will be aorta. Okay, so from aorta basically the heart, that is oxygenated blood is getting supplied, then it will branch. So let's say this is heart now. Okay, so here is the heart. So from heart what is happening? There is aorta which is leaving. Okay, so this aorta which is there, so or the artery, so this is going to divide. Okay, so where it will branch, and it is going to two of the kidneys. Okay, so renal arteries we call them. Okay, so here one side one kidney is here, on the other side there is another kidney. Okay, so these kidneys what are there? So they will be receiving this blood. Okay, so that is renal artery will be received by this one. So in this case this we call as a renal artery as it is coming, and you know this particular artery what is there? So it will divide multiple times to form the arteriole, because there are 1 million nephron which are present here. Okay, so 1 million nephron here and 1 million nephron here. So what will happen? this artery, renal artery, will divide 1 million times, so that afferent arteriole is supplied to each one of these nephrons. Okay, so it will divide 1 million times. Okay, and what will happen, so each of the nephrons which are here, okay, so all these nephrons are going to get this afferent arteriole, then there will be capillary formation and then efferent. So same way this will be formed. Okay, so that means 1 million times this artery has to divide, and everywhere it has to supply this afferent arteriole. So through this, whatever blood is coming over here, so that blood is actually flowing with a particular pressure. Okay, so now here as the blood comes, okay, so as the blood comes, so blood is going to enter, and you know here the narrow diameter is there, and here also in the afferent arteriole also there is a narrow diameter. And when we talk, usually what happens, so whenever the volume is less, okay, so volume, okay, sorry, so whenever volume is less, what will happen, the pressure, okay, so pressure is gonna increase. So here in this case, since the volume is less over here, okay, so what is happening now? There is some camera disturbance I have today. Let me just check. Okay, let me just check if it is fine now. What happens, so whenever the volume is less, okay, so volume, okay, sorry, so whenever volume, just check if it is fine now. What happens, so whenever the volume is less, okay, so whenever volume, this is still the same, no? I don't know what's the problem. Alright, anyway, I'll continue as such only for today. Let me check it for the next classes. Okay, so anyway, I am visible, my diagram is visible, so I hope it's fine as of now. Anyway, I'll continue as such only for today. Okay, so yeah, 1 second. Yeah, so now you know whenever there is a decrease in volume, the pressure is gonna increase. Okay, and as you can see here, so here the diameter was somewhat wide, and as the blood is gonna enter this capillary, so glomerulus is basically the capillary, what will happen now, this blood, so since there is a narrow diameter, so most of the blood what is there, mainly not the blood exactly, the liquid part of a blood what is there, so that is gonna enter into this Bowman's capsule. So what is happening, the filtration is occurring. Okay, so the blood here as the pressure has increased, okay, so liquid part, I'll just change the color so that again you can make a differences with respect to that. So maybe I can take the different color now. Okay, so here you know, so most of the liquid part of a blood what is there, so that will all get filtered in here. Okay, so you will here this Bowman's capsule is gonna receive the liquid part. Okay, and that liquid part of a blood what is there, so that we call as a glomerular filtrate. Okay, so it is glomerular filtrate. All right, so this glomerular filtrate what is there, so this is actually not the urine, so there is some more process that has to occur. Okay, so now why do you think this particular liquid part is getting, or I mean entering the Bowman's capsule? So that is mainly because of the hydrostatic pressure which was generated as there is decrease in volume and increase in pressure. So what is happening, so there is a hydrostatic pressure which is built. Okay, and because of that the blood part of, I mean liquid part of a blood what is there, so that will get filtered over here, Okay, so that filtered part which we call as a glomerular filtrate, and this glomerular filtrate, what is there? So it is actually not the urine. Okay, so there is some more. So this glomerular filtrate will have the useful compounds for us. So there is glucose might be there. Okay, so some amount of glucose is required, then some amount of water is also required, because the blood which is leaving through the efferent arteriole, it will be thicker. Okay, and the blood here which was there, so it was of a normal same concentration, like whatever normal concentration of blood has to be there, so that was the concentration. But here when the blood is leaving after the filtration process, that is after the ultrafiltration, so this process, what is there? So this process we mentioned here itself, I'll write. So the process is known as ultrafiltration. Okay, so why do we call it as ultrafiltration? That is mainly because so most of the liquid part is getting filtered over here. Okay, and the thicker blood, okay, so the blood which is leaving through this particular efferent arteriole, so the blood will be thicker here. Okay, so we need to maintain the same concentration of the blood, so that is why the required elements like glucose, water, so these are all gonna get reabsorbed. So this particular glomerular filtrate, when we mention, so it will have salts also. Okay, so it will also have a urea and other things. So basically urea and all the unwanted compounds, what are there? The excess amount of salt. So let's say there is excess amount of salt, so this we need to get rid of, and urea we need to get rid of, and also some amount of water we need to get rid of. Okay, so for all these conditions, so what has to be done? So in order to get rid of unwanted things, so basically the filtration is occurring. Okay, but there are some useful compounds also, like glucose is there, water is there, or it can be vitamin certain cases, and there can be some proteins also, we require them. So here in this process, basically once the filtrate, once the glomerular filtrate enters here, okay, so you can see the glomerular filtrate has entered, and also you can see this particular efferent arteriole, what is them? So this has divided many times, and it has formed a capillary here. So all, like most of the processes, basically when we mention, so most of the wanted things, like the ones which are required in the blood, so glucose is required, the water is required, so here the reabsorption process will occur. Okay, not everything is reabsorption. One second. Alright, so reabsorption process will occur. That means only the ones which are required in the blood, so only that will get reabsorbed, but not the everything. So what will happen here? So urea is not getting absorbed, then excessive salts what are there, so they are not getting reabsorbed here. So only the water, glucose, there might be vitamins, so which are required, so only that will get reabsorbed. Okay, and that is why we are mentioning, or we are calling this particular process as selective reabsorption. Okay, so we call it as a selective reabsorption. That is because not all the unwanted things or anything, so it is not getting absorbed, only to maintain. So here also water is getting reabsorbed, again not full water is getting reabsorbed, only the amount of water that is required in order to maintain the same concentration of blood, so only that amount is getting reabsorbed. That is where it is occurring. So in the PCT, so majority of the reabsorption will occur in PCT. Then in Henle's loop also you will find reabsorption, the selective reabsorption itself. Then to some extent here in the DCT also. Okay, so in DCT also to some extent you can find the reabsorption occurring. Okay, so now Henle's loop when we mention, so here also there is reabsorption which is occurring, so that is why you can see this. Now let's say this liquid part is entering, that is glomerular filtrate is entering over here also. Okay, so whatever amount of water is required or the glucose is required, so through the diffusion, so here this liquid part, what is this? So it will get into this capillary. Okay, so there is exchange that is going to occur between these capillary and then this column. Okay, so now this Henle's loop is there, so here the water is there, so some amount of water is going to get into this capillary. So here also it is the same thing, some amount of water, the glucose, etc., so they'll all get reabsorbed here into this capillary. Okay, so capillary is reabsorbing the water and glucose, that is mainly because so it has to maintain the same concentration. So we don't want to lose the every part of a, I mean every liquid, or the liquid part which is of a blood, so we don't want to lose it. Okay, so that is why everything is getting reabsorbed. Exception is that the nitrogenous waste or any excess amount of sauce, so we want to get rid of them, and along with some amount of water, so that will be gone. Okay, so that will be removed out in the form of a urine. And here when we compare it with respect to DCT, okay, so what will happen in the DCT? So one thing is reabsorption is occurring, and here also you can see to some extent the reabsorption occurring, and to some extent here also you can see the reabsorption that is occurring. And now in this case the DCT, okay, so mainly the tubular secretion is gonna be seen. So now when we mention about the tubular secretion, okay, so what exactly is tubular secretion here? So this part, okay, so let's say you might not be keeping well, so you are consuming excess amount of medicine, so basically drugs what we call. Okay, so if you are consuming excess amount of these drugs, so what can happen? So unwanted or excess amount of vitamin also we can consider, so all the unwanted materials or which are in excess, so here the cells what are there. Okay, so this DCT will be made up of a cells again, so this everything is made up of a cell. Okay, so these cells what are there, so they will be secreting what, so they are going to secrete a excess amount of drug. Okay, so that is later on gonna join, so that is tubular secretion. So this is a tube now, and the tube cells what are there, so they'll be secreting the drugs. Okay, so drug secretion, or it can be vitamin secretion. Okay, so basically all these can get secreted over here, that is through the DCT. And then after the tubular secretion process, so whatever amount of liquid what is there, so here this liquid is gonna enter. Okay, and it will be flowing into the pelvis area of a kidney. Okay, so this will be entering the pelvis area of a kidney. So now this one, the one which enters, so after the tubular process, I mean after the tubular secretion, the fluid that is entering, so that fluid itself we call as a urine. Okay, not until this one. So after tubular secretion, so first is ultrafiltration has to occur, so we get the glomerular filtrate, reabsorption has to occur, and then the tubular secretion has to occur. So only after that we call it as a urine, not until then. So until then we will be calling it as a glomerular filtrate itself. Is this clear? I hope you all are listening to me, because chat is anyway not available for me. Okay, so I just hope that you are all understanding. Okay, so now here this is the same thing. Okay, what we discussed. So we discussed about the formation of glomerular filtrate. Okay, so this will consist of water, urea, salts, glucose, and other plasma solutes. Okay, so thicker part of the blood which is left behind in the glomerulus after ultrafiltration. So there can be corpuscles, that is RBC, the WBC will be there. So there are certain proteins which will be there. Okay, just a second. Okay, so there will be certain proteins and large molecules which are there, so basically larger proteins only. So those are actually carried by the efferent arteriole, and it is thicker. So now we need to maintain the same concentration, so this is actually the one which is thicker. So in order to maintain the same concentration, the reabsorption has to occur. So glomerular filtrate entering the renal tubule is not the urine, it is extremely dilute solution which is having usable materials. Okay, so so including the glucose and some salt. So let's say excess amount is there, anyway it will be thrown out, and some salts like sodium if it is required, so this can be taken down. Okay, so but here the reabsorption what we mentioned, okay, so this is only to an extent that the normal concentration, this is what I was highlighting, so normal concentration of blood is not disturbed. Okay, so only to that extent the water is going to get absorbed, the glucose is going to get absorbed, so all this absorption what is occurring back into the capillary, so that is mainly occurring with respect to, in order to maintain the normal concentration of the blood. Okay, and that we mention as a selective absorption. So here you can see, so I have mentioned about the selective absorption, so this is actually the reabsorption itself. Okay, so selective absorption is occurring, so only to maintain a normal concentration. Then there are certain substances like potassium in the normal course, or a large number of foreign chemicals what we talk, or there can be drugs like penicillin, so these are passed into forming urine. Okay, so we discussed about the tubular secretion also. So there is, let's say you are consuming a medication. Okay, so in those cases, under those conditions also, so your DCT what is there, so it will be secreting the drugs into the forming urine. Okay, so now the passage involves the activity of the cells of the tubular wall. So let's say this is a DCT now, so here the cells what are there in the DCT, so they are responsible for secretion of this penicillin, or excess amount of potassium will be secreted and it will be thrown out. Okay, and that is why this is a tube now, and tubular cells what are there, so they are secreting, so that is why the name is given as a tubular secretion. So filtrate which is left after reabsorption and tubular secretion, so now we can call it as a urine. Okay, so what exactly happens, so we will see this part. So we have already seen, so in glomerulus there is ultrafiltration which is occurring. Then the Bowman's capsule when we mention, so it is going to receive the glomerular filtrate. Okay, so proximal convoluted tubule when we mention, so this will absorb the water, the reabsorption that is about 2/3. Then glucose reabsorption, the sodium and the chloride ions are going to reabsorb, some absorption of water also we can see, and the sodium ions in Henle's loop we can see. Okay, then there is reabsorption of remaining chlorides and some water will occur here also. Okay, then the walls of which are there, that is DCT, so they will be secreting the potassium and foreign chemicals such as the penicillin and other drugs. Okay, so those will be secreted, and then we can call, so after all these processes itself we can call it as a urine. Okay, so final urine what is there, so it will be passing into the collecting duct, then from collecting duct it is going to the pelvis of the kidney, then through the ureter, so from pelvis it will enter the ureter. Okay, then from ureter it is going to enter into the urinary bladder, so that is by peristalsis, that is wave like motion with respect to constriction. So basically this particular ureter what is there, so it will show a wave like movement in the ureter, so that the urine can flow easily, and it is also due to the gravity. All right, so urine is expelled from the urinary bladder, so basically there will be a urinary bladder. Okay, so from urinary bladder the urine will be expelled, that is by relaxation of the sphincter muscles. So you know there will be sphincter muscles which are located at the opening of the urinary bladder into the urethra under the impulse from the nervous system, so such a process. So here, so let's say this is a kidney now. Okay, so this one is now a kidney. All right, you know there will be this pelvis area. Okay, so there are pyramid like structures what you find. Okay, so there are these pyramids which are present. So now this collecting duct, so pyramid is basically, it will be made up of Henle's loop and then the collecting ducts itself. So now this collecting duct, okay, so it is going to empty the urine into this pelvis area itself. So from here it is getting into the ureter. So this one was actually the pelvis. Okay, so this part what is there, so this is pelvis of a kidney, this one is ureter. All right, and here let's say this is now a ureter which is somewhat projecting as such, and then there is urinary bladder over here. Okay, so this one is a urinary bladder. So from the other kidney also it is the same thing. Okay, so now this one is urinary bladder, and so the in order to guard the opening and closing of the urinary bladder, so there are sphincter muscles over here. Okay, so these sphincter muscles will guard the opening and closing of this particular urethra. Okay, so now here what you find, so this is actually the urethra. Okay, so this is urethra, and these are actually the sphincter muscles. I hope it is clear for you all. So this one is sphincter muscle. Okay, so this is under the impulse from the brain, so this is gonna open up, and when it open up, one is gonna urinate, and the urination process, what is that? So that we call as a micturition or a urination also. Okay, so you always you need to know the other term also when you are studying. Now all these functions involved in urine formation, what are them? So they all require the energy. Okay, so oxygen demand to the kidney is, so our artery is coming. Okay, artery, I mean aorta, which was there. So aorta will divide. Okay, so it will be dividing, so it will be further dividing to supply to other organs also, but this aorta is dividing into two branches basically whenever it is going to the kidney. Okay, so here the right kidney is also going to receive this blood vessel. Okay, and the left kidney, what is that? So that is also going to receive its blood vessel. So here the kidney is there, and here one more kidney is there. Okay, so here the blood supply will be done through the aorta or a artery which is branching, and it is supplying a blood vessel to the kidney. So why this is happening? It will be carrying the oxygenated blood, and the oxygen demand is much higher in case of this one, that is in case of kidneys. So kidneys, it is about 6 to 7 times higher than what is required by the muscle. Okay, so that is why there has to be a supply of oxygenated blood to the kidneys. Okay, so kidneys always need a large amount of oxygen for their working, as they are removing a waste. Okay, now when we mention about the color of a urine, so this will be clear yellow due to the pigment urochrome. Okay, so this is gonna vary with the diet also. Then volume, when we mention, so it can be about 1 to 1.5 liters per day, but it is gonna vary again. Okay, then pH, when we mention, so it is 5 to 8, but again here, so usually approximately we will consider it to be 6 itself, which is again a acidic urine. Okay, so urine which is produced by our body, so this will be slightly acidic. Then the protein which is mixed in it. Okay, so protein diet make when let's say you are consuming more of a protein, then in those cases you can find it to be more acidic. Okay, while the vegetable diet, when you consume, so it will make it alkaline. Then odor, when we mention, on standing the smell of urine becomes strong ammonia like due to bacterial activity, otherwise we can find the faint smell. So this one is a specific gravity now. All right, 1 second. Okay. Okay, so constituents of urine we will see. Okay, so normal human, when we mention, so basically these all compounds will be there. So this is organic, and this one is inorganic. So urea can be about 2 to 3. Okay, so 2.3 also. So this is actually 2.3. Okay, then there is creatinine 1.5, then uric acid 0.7, the others what are there, so 2 to 2.6. Then sodium chloride, so these are all inorganic ones, so there is 9, potassium chloride 2.5, ammonia 0.6, and others, so this is 2.5. so you will find find urea also, creatinine also, uric acid also, others also. Okay, then there is sodium chloride, there is potassium chloride, then there is ammonia also. You want to remove ammonia, then you need to drink about 500 ml of water. That is to remove 1 gram of urea. Okay, so to remove 1 gram of urea, I mean sorry, not the urea, ammonia, 500 ml is required. So let's say you want to remove 1 gram of urea. Okay, so in order to remove 1 gram of urea, okay, so I think like not I think it is basically 50 ml. Okay, so 50 ml of water is sufficient. Let's say we want to remove 1 gram of uric acid, and for this usually 10 to 15 ml of water is sufficient. Okay, so so that is why this ammonia, when we mention, so this is actually highly toxic. All right, this is again toxic itself, so this is also toxic. And when we compare uric acid, so this is somewhat less toxic compared to both of them. Okay, so here then abnormal constituents of urine, when we mention, so blood passes with the urine. Okay, so that is due to infection in the urinary tract or kidney stone or a tumor. Okay, so these are the abnormal ones. So blood cells, when we find, so they'll ask you this question. Okay, so keep it in mind, keep this one in mind, what exactly is hematuria. So basically the blood is going to pass along with the urine. Then glucose or glycosuria, when we mention, so excess amount of glucose is going to pass through the urine, and that we mention as a condition which is a diabetes mellitus. Okay, then here in this case, so this you already know, so it can be because of the insufficient insulin as well. Then when tasteless, so let's say your urine is tasteless, then in that case it is due to the much water in the urine. Okay, so there is much more water in the urine, which is called as a condition diabetes insipidus, due to insufficient ADH hormone, antidiuretic hormone, if it is not present in a proper or insufficient amount. Basically in a regular concentration, if it is not there and it is insufficient, then it will lead to more of a water. Okay, so it will be more watery and it will be tasteless. Okay, so the urine, okay, so that we mention as a diabetes insipid. Insipid means it is basically the tasteless. Then albumin, so due to high blood pressure or due to increased permeability of the Bowman's capsule membrane on account of bacterial infection. So when we mention about the albumin, so basically it can be due to blood pressure or due to increased permeability of the Bowman's capsule membrane on account of the, so if there is bacterial infection, so regular osmoregulation cannot be done. So we will see osmoregulation. So albumin is going to help in osmoregulation, that is in maintaining the same concentration of blood. Okay, so that will be done through the, so it is same concentration with respect to water and minerals also, so that will be done through the albumin itself. Okay, so here this albumin is going to do this one, so we will see this. Then the bile pigments, so anemia, so if there is if the RBCs are getting dyed, okay, so it can lead to the formation, or we can find bile pigments in our urine. Okay, so then there can be hepatitis and jaundice, what we call, or due to the liver cirrhosis, that is cells of a liver are getting dyed. Okay, so regulation of urine output, when we mention, so it is under the control of pituitary gland, and diuresis is basically increased production of the urine, so that is concentration of the urine water reabsorption. So this is basically by a ADH. Okay, so if this ADH is insufficient, then what is going to happen? So ADH secretion is reduced, that is insufficient ADH is available, so in that case increased production of urine we can find. Okay, and this is called as diuresis. So this one is again important. Okay, so this is again, so you keep this one in mind. So basically diuresis, when we mention, so increased production of urine we can see. Okay, so that is mainly because of the ADH. So substances that increase the formation of urine are called as diuretic. So example, there is liquid diet, or it can be tea, or it can be coffee, alcohol, etc. Okay, so all these can contribute in the urine formation. Then when we mention about the gout and the kidney stone, so uric acid is relatively less soluble, okay. So this is less soluble in water and it is going to become crystallized and it will get deposited in the joints causing the gout, okay. So in those cases excessive uric acid and certain salts like calcium oxalate or maybe the other source of kidney stones when the kidneys are not working fully normally, okay. So uric acid is the one which is accumulated at the joint, okay. So it will get deposited at the joint, that is mainly because it is less soluble in water, okay, and it will crystallize and it will get deposited in there. One second, let me just check what else is there so that yeah, I'll cover this chapter today itself. All right. Yeah. Okay, now osmoregulation. Okay, so now when we mention about the osmoregulation, the kidney while removing the waste. So what is happening? You know, kidney is mainly responsible for removal of the waste. Okay, that is like urea from the blood. Okay, so this is basically excess amount of salts are also removed, vitamins, or there can be certain drugs which are removed. So here in this case, so from the blood, so this is also going to regulate its composition. So composition regulation is done, so that is the percentage of water, so that itself we call as a osmoregulation. That means amount or whatever the percentage of water is required or certain salts are required, and that same concentration of a blood will be maintained, so that is by absorption, okay. So that is reabsorption. So same concentration of blood has to be maintained over here, okay. So this function is called as osmoregulation, so the regulation of the osmotic pressure of the blood. So you can also mention this one, okay. So regulation of osmotic pressure of the blood we can mention as a osmoregulation. So drinking enough water directly or through food helps the kidney in their proper working. So in tropical climates as in our own country, okay, so we drink a lot of water during the summer, so yet we urinate fewer times. So this one give a reason you will get, okay. So this you need to be very like careful, you need to write the same answer over here. And you know, the previously we studied the afferent arteriole and the efferent arteriole, and they'll ask you why the efferent arteriole is having a thicker blood, okay. So there also give a reason type question you they can ask. So here the urine pass generally is thicker in case of a winter, okay. So the reason okay, just a second. So the reason is that in summer what will happen, so we lose a considerable part of the water, that is through the perspiration process, okay. So perspiration process, what is this? So that is going to help in losing the water, and the kidneys have to reabsorb more water, okay. So kidneys, what are they? So they'll be absorbing more water from the urine, making it more concentrated, okay. So perspiration is basically in the form of sweating, okay. So sweating, and here in this case kidneys will be reabsorbing more water also. So that is why the urine which is there, so that will be more concentrated in case of a water, okay. I mean in case of a urine. So yes, just a second, let me just drink water. So is this clear? Any doubt you have till here? Okay, so in cholera, okay, so cholera patient, what happens? The patient suffers from vomiting and also watery bubbles we will find, you know, loose motion it is basically, okay. So his intestine, the person who is having a cholera, so you know, in the intestine basically absorption of water occurs. So his intestines are unable to absorb water into the blood, okay. And because of that, what is happening? His kidneys is, okay, so result is that his kidneys reabsorb almost all the water from the urine. Okay, so what will happen? So most of the water what is there, so that is kidney is gonna absorb most of the water from the urine in the renal tubules, and with it even the urea. Okay, so basically absorption will occur. So ultimately the patient is gonna die due to poisoning by the accumulation of high quantity of urea in his body. Okay, so that condition we call as a uremia. So this is why is it happening? It is basically the intestines what are there. Okay, so intestine is not absorbing enough amount of water, and so kidney is going to absorb the water here. Okay, so here you can see that almost all the water from the urine in the renal tubules, and with even the urea. Okay, with it even the urea, so that is basically going to be absorbed. Then immediate treatment is to replenish the water in the blood, that is by glucose saline drip or by giving oral rehydration solution, that is through the mouth. Okay, so immediate treatment is, so let's say so they are continuously vomiting and there is loose motion also, so in those cases one, so that is basically to replenish the water in the blood. So in order to maintain the amount of water in the blood, that is by glucose saline drip or by giving oral dehydration solution. So basically not dehydration, rehydration solution, so that, so what is going to happen, so same amount of salt and water can be maintained in the blood. Okay, okay, so water balance in human body when we mention, so adult human body will have about nearly 40 liters of water. Okay, so in that which forms 60% of the body weight. All right, so this is in a human body. So now in urine you can see this, so urine it is about 1500 ml, then in sweat it is about 500 ml, in breath 400, and in feces 100, so total of 2500 ml. Okay, then gain when we mention, so in drink as water or in beverages in food, so 1500 if we are drinking, in food it is about through 700, then metabolic water, so it is about 300. Okay, so intake and output of a water would slightly vary, that is according to the climate and the lifestyle of a person's. Okay, so this is adult human body contains about 40 liters of water will be there, which will form about 60% of the body weight. Now average daily loss and gain of water in an adult human male in good health and rest in temperate, one second, at so this is in good health at rest in temperate climates. Okay, so this is the condition, so this is going to vary based on in what kind of a lifestyle they are having. Okay, so other than that there is information, the miscellaneous information we will check. So here the artificial kidney, so you might have heard or maybe you might have seen any of your relatives, maybe your neighbors, so they'll go for the dialysis process. Okay, so dialysis process we will see what exactly happens. Okay, it is just to remove the urine which is formed in your body as the kidneys are not working properly. Okay, so it is an artificial kidney. So if one kidney is damaged or removed for some reason, okay, it can be any reason, the other kidney alone, okay, so one kidney alone can perform this function. Okay, so it is sufficient for the excretory needs and the person can lead a normal life with the one kidney itself, but failure of both the kidneys would lead to death. Okay, so that is why artificial kidney is a dialysis machine. Okay, so here the patient's blood is led from the radial artery, so it is led from the radial artery in his arm through the machine. Okay, so that is through the machine only. So what they are going to do, so they are connecting this blood vessel, that is patient's blood is led from the radial artery in his arm, that is through the machine where the urea and the excess salts are removed. Okay, so if there is any excess salt and urea is present, so that can be removed through this arm blood vessel itself. Okay, and the purified blood what is there, so it will be returned to a vein in the same arm. Okay, so in case of permanent damage to the kidneys, dialysis is to be repeated for about 12 hours twice a week. So let's say, so in there is a permanent damage, so kidneys are not working at all, so dialysis has to be repeated for 12 hours. So 12 hours it has to happen. Okay, so that is twice a week. So in 1 week, 2 days. Okay, so 2 days completely they need to go for this particular. Okay, so days is basically the not the night. All right, so 12 hours means it is a long process. So 12 hours they need to purify their blood. So now here with respect to the same, so summary of excretion we will see. So we have seen all these in the previous classes. So if you haven't watched the session, so you can just rewatch. You can recording will be available, so you can watch these recordings over here. Okay, so any doubts you have, you can ask in here. All right, so carbon dioxide when we mention, so basically excretory organs, so there are lungs. Okay, so lungs, so through the lungs only the carbon dioxide removal is done, so as a gas in a expired air. Okay, then there are mineral salts there. Okay, then there is nitrogenous waste products, so kidneys, so as constituents of urine. Okay, so mineral salts when we mention, so or the nitrogenous waste product, mainly the urea, the creatinine, and then there is a uric acid. So all these can be removed through the kidney, also to some extent skin also. Okay, so this is basically the constituents of urine. Skin when we mention, so it can be through the sweat, where the sweat is going to contain small amount of nitrogenous waste. So you know, sweat gland when we mention, it is actually not the excretory organ, because the sweat production is occurring, so that is to cool down the body, because surrounding temperature is too hot, and because of that your body is also getting hotter. Okay, so to avoid that, that is in order to cool down your body, the sweat production is occurring. Okay, so that is why sweat we cannot consider completely as a excretory function. It is basically to maintain the temperature of your body, so sweat production is occurring. Okay, so now moving on to the other one, so water. Okay, so water, how it is removed? So one thing is through the kidneys. Just a second. Okay, so one thing, so it can be removed with the help of kidneys also. Where is the cursor? Okay, here. Okay, so with the kidneys anyway, in the water, I mean in the form of urine itself, so excess water, so that is excreted out as a urine. Then skin, so in the skin it is basically through the sweat. Okay, so there will be formation of a sweat. And then when we mention about the lungs, so water is lost as water vapor. Okay, so it will be lost as water vapor in the expired air. Okay, then when we mention about the liver, so that is through the intestine. All right, so through the intestine the water will through the lungs also. You can just hold in your hand as such. All right, then you can see that you can feel some amount of moisture, or some there is a vapor that has been released, so you can feel that. Or maybe you can just blow the air on the glass also, you will find this smaller water droplet, so that will prove that so lung is also losing some amount of water through the in the form of a water vapor. So it is the lung. Now coming on to the liver, so I'll just explain about this one. So I said something over here. So bile pigments, that is from hemoglobin breakdown. So you know, RBCs when they die, okay, so there will be this hemoglobin which is present on RBC. So let's say there is RBC now, which is a flat one or a disc structure. Okay, and on this one, so you will find this hemoglobin which is present. So this hemoglobin will be broken down, so that is breaking down is gonna occur at the liver. Okay, so through the intestine, that is basically once the bile pigments are formed, or the once the bile juice formation occurs, so this is gonna open through the duct into this intestine. Okay, so this will be opening into the intestine, and you know, so that is via bile juice poured into the duodenum. So that means so intestine, mainly the duodenum, it is going to open up, and the yellow coloration of a stool or a fecal material what you find. Okay, so that yellow coloration of fecal material is mainly because of this bile pigment which is released into the intestine. Okay, so that is a breakdown product of, so when hemoglobin is broken down, so this is gonna form the bile pigments. Okay, so this is broken down and there will be formation of bile pigments what we can see. So it is actually the bilirubin also. Okay, so there is bilirubin and then there is biliverdin. Okay, so these are being produced. Alright, so with this we have finished our chapter. So any doubt you have, you can ask in your doubts. Alright, so before I end the class, let me just say about this. So Unacademy subscription features. Okay, so these are the Unacademy subscription features. One second. Alright, so basically the benefits of this one is you can learn live from the comfort of your home. Okay, so unlimited access to all the courses you'll have. Then top educators of India are gonna take the classes for you in one platform. Then there is a regular doubt clearing session. So what exactly is this? So after about every 3 sessions, one session we will be keeping, and this one session is a doubt clearing session, or we can mention it as answer writing session, where you can clear your doubts with respect to the topics that have been covered in the previous 3 sessions. Okay, so this is all about the doubt clearing. Okay, then there is exhaustive coverage of a syllabus will be done. The mentorship and guidance is given, study materials is given, practice tests, live test series, batch courses. So you will have benefit of all this, and all you need to do is use the code VANIYT in order to get 10% discount on your subscription. Okay, so with this code, so this code is going to help you in getting 10% discount, and also with that you can also get like daily practice session, the weekly mock test series, live quizzes, daily MCQ and subjective test series. So you can take up these. Okay, so you can enter my educator code, so here to get 10% discount. Okay, so let's say you want a 10% discount, so this is my code which you can enter over here, that is VANIYT. All right, so let's say you are going for these many months of subscription. All right, so on top of, so you are already getting save 64%. So basically, so if you are going for a higher number of or higher months of subscription, so the cost is gonna reduce, as you can check in here. Okay, so you can go for the subscription based on your convenience. Okay, and with that you will anyway be getting 10% discount when you use my referral code. Okay, so this is actually the ongoing batch now, which has started on 17th of Jan. Okay, so Udan batch, the delivery of languages Hindi. So let's say you are taking a subscription for class 10, so class 11 to 12 subscription, it will be absolutely free for you all. Okay, so it is offer. So same way the Mavericks batch which is there, so this is also all subject batch, and the delivery of language is basically English over here. And you know, so this is also started, that means it is again an ongoing batch. Okay, so you'll have access to this one if you have taken a plus subscription, and for that you can use my code VANIYT. All right, now moving on to the next one. So this is a completed batch, so here you can get the recordings. Okay, so you can watch the recording if you have already taken a subscription. So here also you can watch the recordings. Okay, so this will be available. All right, and I'll be conducting the special classes. Okay, so in special classes I'll be giving the poll, I'll show you your competitors, so you'll be getting first, second and third position. Okay, then also during a live class you can type in your question, or you can attach an image and you can send it, and the educator who is taking your class, so they'll get a notification related to the same, and they'll be clearing your doubts. So how exactly, like how do you watch my special classes? That is on Unacademy. So all you need to do is download the Unacademy Learners app. Okay, so then click on get started. Okay, so you can click on get started, then you can enter your mobile number and a OTP, then you can sign up using your email ID. Okay, so what goal are you preparing for? So since you are in class 9 and 10 basically, or 11 and 12, so you can select this particular section. So select class 6 to 12, then you need to choose your class. Okay, so there you can choose, so this option will also be available, so you can choose ICSE or ISC class 9, 10, 11, 12, what you are in. So since now you are in 10, so you can choose ICSE 10. Okay, then you can also add other goals over there. Then other than that, this one's the languages. Okay, so you can choose any of the languages, the preferred language in which you want to learn. Then go to the description of this video and click on the special class link. Also you can click on notify me, and you will get 10 minutes before the class is gonna begin. Okay, so you'll be getting a notification. And also you can use my code again, Vani YT, to unlock this special class for free. Okay, so download the Unacademy learners app now. Okay, so there are actually, so try all of these features. Okay, so all of these features you can try if you have taken a plus subscription. And to take the plus subscription, so you can go for any of the months of subscription based on your convenience. And with that, if you are using this code, you will be getting 10% discount. Okay, it is Vani YT by name. All right, and also with this code, okay, so with this code you can unlock many free products. Okay, so there is unlock option, so this unlock many free products which are available on your on the Unacademy learners app. So with this you can unlock them as well. Okay, so see you all in the next session. So Unacademy, let's crack it. See you all. Bye bye. Thank you so much.